The next talk is Understanding the Consequences of DNMT3A Disruption from Molecular Mechanisms to Organismal Impact, and it will be presented by Dr. Harrison Gabel. Dr. Gabel received his BA in molecular biology from Princeton University and his PhD in genetics from Harvard University. He is currently an associate professor in the Department of Neuroscience at Washington University in St. Louis. Since establishing his laboratory in 2015, Dr. Gabel's research group has studied molecular mechanisms of gene regulation that contribute to development and plasticity in the mammalian brain. The lab combines genetic, genomic, and biochemical approaches in mouse and human models to identify and dissect important gene regulatory pathways in neurons. A broad goal of this work is to understand how disruption of transcriptional regulation can lead to autism and related neurodevelopmental disorders. Dr. Gabel's research group has developed multiple preclinical models of TBRS. In ongoing studies, his group is investigating the molecular and cellular consequences of DNMT3A mutation in these models and developing experimental systems to evaluate candidate therapeutic approaches for TBRS. Please welcome Dr. Gabel.
Well, thanks very much. It's really an honor to be back here in person and actually seeing some people in person for the first time. Really great to have this opportunity to share scientific ideas and then pair it up with a family meeting so that we can engage with the community. So thanks very much. So I'll be telling you about our work that sort of hopefully is bridging molecular to organismal understanding in TBRS. But I'll tell you a little bit about the origins of how my lab got into this and then how we're working on TBRS models. So my lab really, again, thinks about molecular mechanisms in the brain, and we study novel molecular mechanisms in the brain that end up intersecting with DNMT3A biology. We really founded the lab based on looking into DNA methylation and how it regulates gene expression in the brain. And of course, as this audience well knows, DNA methylation is catalyzed by the DNA methyltransferases. But what wasn't appreciated by the research community until, I guess, not so recently now, a few years ago, is that DNA methylation, which largely occurs at CG dinucleotides in most tissues, is actually relatively unique in neurons in our brains and the brains of mice and other mammals, where there's a high degree of non-CG methylation that's occurring. So methylation primarily at CA dinucleotides. This is something that emerged just sort of as I was finishing my postdoc, right as I was about to start my own lab, and we were really interested in studying how this non-CG methylation functions, because it turns out that about half the methylation in neurons is actually this methyl CA. And this wasn't appreciated until quite recently, and to us seemed like it was probably pretty important for the function of the brain if there was so much of this methylation. And in fact, we got to studying how this methylation worked because we found that the Rett syndrome protein, MECP2, actually binds very tightly to this methylation. In this little cartoon, you can see these lollipops as methyl CA, and MECP2 we found bound very tightly to it, and we started studying how this methyl CA and DNA methylation in general helped regulate genes, particularly through enhancers, sort of cis-regulatory elements in the genome. And so we were established and pretty good at studying this phenomenon in the brain and wanted to know how the deposition of this methyl CA occurred, and DNMT3A was the best candidate for a number of reasons. So we and others looked into this and found that if you knock out DNMT3A, you can totally block the buildup of this methyl CA in neurons, shown here for a conditional knockout experiment using a Cree driver only in post-mitotic neurons. So the methyl CA levels are really gone in these. This is the background level of the assay effectively. So you have no methyl CA in neurons if you knock out DNMT3A. And this was just a little bit after DNMT3A was identified as mutated in TBRS. And so we felt like we were in a really good position to understand not only the basic mechanisms of how this methylation goes in and is read out, but how it might be affected in these disorders. And so we initially really dove into trying to understand methyl CA effects. But actually, as we've partnered with the TBRS community and understand the need to have a broader understanding of the whole disorder, we've moved into looking at sort of all the ways DNA methylation is affected in the brain and also trying to model the disorder more generally, particularly making mouse models. So what I'm going to do, that's just a brief introduction of where we're coming from and what we tend to focus on. What I'm going to do in this talk is tell you a little bit, give you an overview of the findings from the mouse models we have thus far, and try to update you on the latest work that we have. And then I wanted to take this as an opportunity in this environment in particular to give you sort of a preview of various things that we've launched, but really are works in progress. So you can maybe give us feedback on if we're barking up the right tree, or we can germinate discussions on how we would attack these things. So first, an overview of our findings. Kate touched on this a little bit in our discussion during the Q&A before. So one of the first things we did when we started studying DNMT3A was to try to look at what the effects of the various mutations are in DNMT3A, because there are so many missense mutations. And we did a number of functional studies in cells and in vitro that showed that although there were lots and lots of missense mutations, at a molecular level, they seemed to be basically molecular loss of function mutations. So the only effects we could see, rather than sort of neomorphic or other types of effects, was that by and large the activity of the enzyme was reduced. That doesn't mean there were complete loss of function mutations. There's actually a spectrum of the relative loss of function that we see in these missense variants. But what we did find was consistently, if we put these mutant proteins back into neurons, that they were unable to efficiently put in this methyl CA in cultured neurons. Because so many of these mutations seem to be, at least so far, sort of differing degrees of loss of function of the protein, we felt it was really relevant to go make the deletion mouse, and this is a constitutive deletion mouse, and study it as a baseline. As I'll tell you, we think it's really important to go after those missense variants to compare to this, but our first studies of this heterozygous deletion mouse really gave us what we still continue to think is a good model of TBRS, where we can replicate many of the features of TBRS in mice to the extent that you can model human disease in mice. We can see overgrowth, long bone length, increased body weight, and adiposity, as Tim Lay's and Peggy Gabel's lab also showed. Actually, sort of before us on the fat side of things, on the body weight side, we've all seen that together. We see behavioral phenotypes in the mice. Very, very hard to model behavior and cognition in mice at the level of intellectual disability. However, you can have multiple assays for cognitive function or different types of neural systems being affected. And we do see phenotypes in these mice that in some ways could be considered mirroring the disorder. And then we saw pretty big effects on the global methyl CA levels, this neuronal specific methylation, I'll get into that more, and site-specific changes in CG methylation that we could tie to gene expression. So we think this is at least one model that's quite useful for the study of TBRS, and there are individuals that have mutations that aren't far off from this particular deletion, either truncations or complete deletions of the gene. So this is one representative model. But we did want to dive into the different alleles, in part maybe where there's, right now when we don't have enough patients to start doing genotype-phenotype correlations from the clinical data, we could sort of take this bottom-up approach where if we made different alleles, we could see if there's variability in phenotypes based on which missense allele you had. And frankly, if there's so many missense alleles in this disorder, we need to have multiple models of missense alleles so that we can try to unpack if there is a mysterious molecular effect that is driving these missense mutations. So these seem like really useful tools to make, and we dove into that ourselves. Two major things we were looking for were across this diversity of mutations in the mouse, can we find shared phenotypes, shared molecular effects? You could think of this as the signature of the sort of core TBRS phenotype. And then we can look at the differences between the alleles for the sort of bottom-up mouse version of genotype-phenotype correlations and thinking about what drives these differences and maybe why we have so many missense alleles. So we generated a P904 mouse model, the P900L, in part because there's a little bit of, and maybe we were a little naive in this regard, or a little hopeful. What we could see in the clinical data suggested that maybe this wasn't quite as severe. I don't know if that's really played out with the few individuals we have, but it seemed like it was either a typical missense mutation or maybe less severe, because there were some individuals in ASD cohorts who didn't necessarily have intellectual disability. In complement to that, actually, Tim Lay had started studying the R882H variant, which is the 878 in mice, and he was focused on this starting because of its preponderance in AML, and had started characterizing it at the somatic level, and we dove into this at the neurobiology level and have continued on in those studies. And at a molecular level in the cancer biology, it's been shown that this has likely a dominant negative effect. So in part, that's our sort of molecular logic, was this might be more severe. So I'm just going to summarize. Diana Beard has presented on this, a lot of this in the past, so I'm going to summarize a whole ton of work from her, sort of characterizing these different alleles. What she basically found was she could see reproducible long bone overgrowth and obesity and body weight phenotypes across these models, and this was also work done by Amanda Smith and Tim Lay's lab in this mutant in particular in a previous publication. We saw a spectrum of behavioral effects, with the 900 frankly not showing very overt behavioral phenotypes in the mice, but getting more extreme in the 878 with really noticeably different sort of, if you're looking at the mice in the cage, you can see effects in these mice where the 900 look quite normal. So we think this does represent a spectrum of severity in the mouse models that may be underneath what's hard to see within the human populations, and we'll see as more data gets collected. What we found interesting about that is that we could see a spectrum of effects at the level of DNA methylation and gene expression as well in doing these studies of primarily the cerebral cortex in the brain, where the more severe the effects, the bigger the effects at the molecular level. So we're hopeful that this will really give us the molecular underpinnings, the pathways that are driving these different phenotypes in the mice. I did want to sort of give you an update. Again, Diana presented on some of this in the past, and we have a study out on bioRxiv, hopefully coming out soon. We've done additional studies that we think are pretty relevant to some of what we've already presented. I wanted to update you on that. One of the things we found that was a bit disappointing from the standpoint of having a good model was that the brain size in these mice and the skull size in these mice is not enlarged. And this is a really sort of central feature of TBRS, that there's enlarged skull and presumed enlarged brain. And in the mice, we didn't see that. In fact, at the time point that we analyzed the mice by MRI, we saw reduced brain size, which is sort of paradoxical. And we wanted to look at this more closely, in part because the mice were imaged at a very late stage, for mouse studies at least, 25 to 30 weeks. So we went back and we looked very closely by doing MRI studies earlier in development, and actually found that the mice don't show a brain size difference really during development or in early adulthood. So this is the overall volume between the wild type control, littermate controls, and mutants. And you can see these subtle but significant trends in the two mutants at these later time points, but really those trends go away, and there is not a reduction in brain size at 8 weeks of age, and certainly not during early development. We really wanted to be thorough about this, so we also did histology and stereology at 8 weeks to count numbers of cells. And we need to dive into this data deeper to go into different brain regions, and we can do that, but the first sort of look at overall cerebrum size and cell counts really shows no difference in the mice. So we still are this model is still lacking what would be a really relevant phenotype to the human disorder of brain overgrowth. However, it's not showing undergrowth at 8 weeks. Now, there is an age-associated reduction in brain size, and I think this should be something we consider studying at the level of aging. It could be related to the metabolic phenotypes and all sorts of other, it could be quite secondary to the actual mutation, but we think there's a good reason to take a closer look at that, and will need to be done at these later time points. So that's on the level of anatomy, what we've done added. We also have been looking more closely at behavioral phenotypes in the mice, with trying to get better assays for thinking about systems that are more relevant to the autism spectrum phenotype that arises in TBRS. This is very hard to study in mice. There are a number of social assays in mice, which have very sort of weak power and can be difficult to run. And we wanted to get closer to output of specific systems that might be relevant to the autistic spectrum. And an emerging idea in the autism field is that sensory, particularly somatosensory dysfunction, could be a driver of some of the autistic phenotype. And you can measure somatosensory function more quantitatively in mice without having to extrapolate to, is the mouse a social mouse, right? Some of these social pros assays. So we started running somatosensory function assays, in particular textured novel object recognition, where we exposed the mice to two objects that are exactly the same, and then swap one object out for a visibly identical object, but actually it has a different feel. So this measures the ability of the mouse to tell the difference by tactile inputs whether or not there's a different object, and the mice actually prefer the novel object. However, in our models, there's a loss of that ability to tell the difference between the two objects, suggesting somatosensory dysfunction in the models. Interestingly, if we just do novel object recognition more broadly, generally with visibly different objects, the mice in the 900 mice, which we think are less severe and maybe closer to an individual with autism with less severe intellectual disability, do not show an effect, where the 878 mice, again, more severe phenotypes in general, and they fail in this assay. So we think this is additional insight into what's going on with the phenotypes in the mice and this spectrum of phenotypes, and can be used in future analyses of the models for thinking about things like therapeutic interventions. Lastly, I just wanted to touch in the mice on the gene expression effects we're seeing in the underlying cause, the DNA methylation changes. As I mentioned, the DNA methylation differences in the mice vary, and that correlates with the severity of phenotypes. The 878 mice show a subtle but significant global loss of CG methylation by whole genome bisulfite sequencing in the cortex of the mice. And then methyl CA is actually sort of beyond 50% lost. So this really is showing a dominant negative effect. In the 900L, there's a 50% reduction, which is very similar to the heterozygous knockout. At the level of CG methylation, again, the 878 is showing very dramatic effects, many differentially methylated regions in the genome compared to the 900L. And the heterozygous knockout is actually about 50% in between here. So what does this mean for gene expression? Well, there are much bigger effects on gene expression in the 878 mice if we, again, use the cerebral cortex as a model. Many more genes affected and higher effects. What we wanted to ask is, are there shared effects here, and is it just the 878 being a stronger version of the 900, or are there sort of are there distinct impacts? And what we found rather surprisingly is that quite a few of the gene expression effects are not simply overlapping to varying degrees, but you can find distinct effects across the two models. We think, and what's intriguing about that is often the genes that go up, for example, in the 900 are trending down a little bit in the 878. So not just no effect, but going the opposite direction. And we're still trying to understand this. Frankly, it's been difficult to understand, but it does underscore that if you use just one model and you say all those gene expression effects are the TBRS gene expression effects, I think you're going to, that diversity of allele-associated effects might mislead you to some extent. And these are genes that potentially have very important functions in the nervous system for connecting up and for nervous system function. But what we can do is take 3 different models now and aggregate them and ask what the core gene expression effects are, really trying to winnow down and say what is the sort of set of core pathways most likely affected that is representative of the core TBRS phenotype. So if we call these TBRS genes up and down, and we're now sort of scrutinizing this gene list that has important annotations for cell growth and for neuronal sort of extension and projection, as potentially the list of genes we should care about the most in terms of their effects. So I think this is giving us something to focus on in terms of the core molecular pathways in TBRS, and then thinking about how you get diverse effects. In addition, there was one question about branching out and thinking about the other disorders that are related. Dr. Tatton-Brown effectively found in her original OGID cohort mutations in NSD1. We talked about PRC2 complex components, and then you have, obviously, DNMT3A. So we, as molecular biologists, have been staring that list saying, are these connected? Where's the molecular biology that connects them? And you can actually see a lot of biology. I must say I'm confused by K27 trimethylation and where it fits in. Sometimes it seems to be going the opposite direction. Sometimes it seems like it should all agree. Where we saw an obvious connection was with the NSD1 protein, which we just published a paper showing that NSD1 actually can act upstream of DNMT3A in the brain and help to target it to the genome to put in its methylation. So really it's just a linear pathway, particularly in the brain. And other groups have described this outside of the brain. So we asked if the gene expression effects we see in the models of Soto syndrome, NSD1 mutation, that we've been developing, and the Rett syndrome gene downstream of DNMT3A, if we can see any underlying sort of core effects that might be common convergent effects that would be of interest for understanding sort of intellectual disability and cognitive deficits in neurodevelopmental disorders in general. And we can actually see very big, robust signatures. This is a genome-wide analysis, rank-rank hypergeometric analysis that shows a sort of very significant overlap of the genes that are upregulated in general with the SOTOS model and the RET model. So again, we can take this sort of even further winnowed down set of overlaps and think about, what does that mean for central convergent molecular pathology in these disorders? And, it's not as simple as just having a gene list. We're going to have to educate ourselves based on what the presentations of the disorders are, and does this really make sense? But we think it's an interesting molecular space to be thinking. So in the interest of time, I'm going to not summarize what I just said, other than to say we think we've identified both core phenotypes and underlying gene expression, and diverse presentations and distinct gene expression effects that could be driving those. So I think I've got a 30-minute time slot, so in 4 minutes or so, I'm going to try to give you a quick preview of some things that are coming along, and I'd be happy to answer questions. We recently took an opportunity to study bone overgrowth and potential orthopedic issues in TBRS because we have a great collaborative group of orthopedic researchers at Washington University. So we engaged with Audrey McCallendon and her student Austin Bell Hensley, along with Diana, the great student sort of spearheading this work, to start looking more closely at bone, the cause of overgrowth if we can understand it, and then looking at the potential for effects on bone health, because there is quite a bit of reports of orthopedic issues. So we thought, well, again, can we do this bottom-up information from the mouse that can then inform us as we collect data from the humans? What we saw, we confirmed bone overgrowth, and then through micro CT studies, we've seen changes in the quality of the bone in these mice. For example, a thinning of cortical thickness in the femur that's observed in both models. And this is really Austin's work, I should say Austin is seeing. And then this does translate into weaker bone that sort of breaks under less force in assays of bone strength. We think this could be relevant if it translates to humans. We've been looking at, Austin has been looking at growth plates and trying to understand the drivers of overgrowth in the disorder. And eventually we want to get into the cellular drivers in the particular cell population that's affected, in the process that's affected. The growth plates are larger, which correlates with the increased bone growth. One thing that Austin noticed that was quite striking is that in a subset of the 878 mice, there's a big increase in the bone adiposity. So the adipocytes have a much larger area. And we think this might relate to the effects in the myeloid lineage, sort of the mesenchymal myeloid lineage, and relate also to what Peggy Goodell's group has shown for adipocytes in fat mass. It could be similar biology going on, but could actually, in a subset of cases, really dramatically affect bone health, we think. So we're going to be digging deeper into what the drivers of that might be. In the last 2 minutes, I just wanted to briefly highlight, and the great thing is Kim Bua, who's a brand new student in my lab, will be presenting a poster on this, so just send you to her poster. We really don't want to sit in isolation just thinking about the biology for biology's sake, but we want to be thinking about, once we have these models, what can we do to be pushing towards testing therapeutics, right? And so we have a whole set of phenotypes that in these mice that we can see reproducibly across multiple models. Those are the phenotypes we would trust the most as an output of TBRS-like symptoms. Now we want to start modeling therapy. And one place to go, which is not happening tomorrow, but is emerging as potentially feasible, and if it could be done, could be a really dramatic therapeutic to introduce, would be genetic therapies, gene therapies. So we want to be modeling if gene therapy is feasible and effective in a TBRS context. It could be in a developmental disorder that putting the gene back late is too late, and you need to be treating the downstream consequences. In other neurodevelopmental disorders, for example, Rett syndrome, it's been shown that actually you can have a dramatic effect if you put the gene back. So Kim is working on a mouse that basically can turn the gene back on in a Cre-inducible fashion, and she can then activate the gene and see what improves in the mice. So she'll tell you all about it at her poster. Go check it out. Finally, I just wanted to very briefly update you on iPS-derived neuron studies that we're doing at WashU, thanks to the great contributions from the TBRS community. This was really launched because of biospecimen collection that the TBRS community facilitated and that the families donated, and we've been doing this with the help of Tim Lay with the infrastructure to derive these lines in the first place, and then working closely with Chris Kroll at WashU. This is my continued work to try to make all of WashU work on TBRS. And she's part of the Intellectual Disabilities and Intellectual and Developmental Disabilities Research Center at WashU. So we've been pushing ahead to make multiple iPS-derived models of TBRS, either from patient-derived cells that then we can correct to make isogenic controls using CRISPR, or deriving certain alleles directly from human stem cells that we again have an isogenic control. We have focused on making the alleles that we have the mice from, so we could do cross-species comparisons potentially. And really the approach that we've been taking, well, Chris's lab has been taking, Gareth Chapman, a really talented postdoc in her lab, is doing this work, is to do directed differentiation schemes and derived inhibitory neuron lineages, excitatory neuron lineages, and potentially other cell types, and to ask, do they have growth phenotypes or relevant phenotypes, and what's the underlying molecular biology? Interestingly, what Gareth sees in the ventral lineage in the 882 variant is actually an overgrowth phenotype at the neuroprogenitor cell stage. That is not seen in the excitatory lineage. So it could be different cell populations are more susceptible to overgrowth and cellular phenotypes that we'll learn from these studies going forward. We can start to do gene expression studies on these cell lines. We're validating them and sort of getting ahead of ourselves, trying to do mechanistic studies as we're still sort of validating that there is as high quality as they need to be. When we get that done, we'll be able to stand on these. So these are a work in progress. But what we find is that the two mutations, you can see gene expression signatures that separate them from their controls. And the gene expression effects that we see really point towards potentially in the inhibitory lineage, overgrowth followed by premature maturation and an increase in the expression of these synaptic proteins that are inherent in the sort of most mature neurons. We're in the process of doing whole genome bisulfite analysis to understand the DNA methylation that could be driving these underlying effects. And we do see overlap between the genes that are affected and the differentially methylated regions that might suggest a direct consequence of loss of DNA methylation. And finally, Jim Huttner at WashU is a cellular physiologist and works closely with Chris Kroll in the IDDRC to be able to then do physiological phenotyping and say, what's the physiological impact in these cells? And in the inhibitory neurons, where we have the most data thus far, we can see changes in the physiology, including evidence of hyperactivity and potentially changes in sort of the nature of the neurons in terms of their fast spiking activity. All right, I'm way over. Thank you for your patience. I want to say thank you to all the folks who did the work, including Diana Beard, who's graduated from my lab, is going to work at Novartis, has really been the powerhouse student behind most of this work. Thankfully, Kim is here and taking up at least some of those studies and continuing on, and I encourage you to chat with her. She's going to be working in the lab on all this. And then thanks to Austin and Gareth, who've been really the drivers of the other studies. Shiyun Zhang was a remarkable undergraduate student, and of course our collaborators, Tim Lay, who's been very generous with working with us on trying to get going with the stem cells, and Chris Kroll. Thank you.